Okay, so going back to this screen share, there has been this concept of using vaccines basically to create antibodies or to force the body to make antibodies against different drugs. And the reason why antibodies are really useful in preventing the effects of drugs or helping with people recovering from drug abuse is because the antibodies have these specific binding sites that can bind to the drug of choice, whatever the target happens to be, in this case fentanyl, and then the antibodies form this bulky structure that prevents the drug plus antibody like complex from going into your brain, and that prevents the drug from having any effect. And it's very cool, very interesting. Felon, I see you. Have a great night. Thank you so much for the sub. Thank you for your stupid jokes. It was fun having you here. And have a good rest of your week. So let's see. So in this article, this is just a fun little image. It's a computer generated image. This is not a real thing, but they're basically showing kind of this cell membrane, and this on the surface of the cell membrane is an opioid receptor in the brain. And basically when the drug or opioid gets into your brain, it binds to these receptors, and then this causes like it maybe increases your dopamine artificially and causes other fun effects. And so, yes, this is just a graph of the horrible increase in opioid overdose deaths. And so let's say that someone has overdosed on something like fentanyl. So ideally, you could give them maybe like a shot of synthetic, not synthetic opioids, but a shot of synthetic antibodies that kind of go around and soak up the chemical in the bloodstream. This is just showing how much normally, like on over here we have heroin, and then we have some other drug, carfentanil, which I don't know much about, and then we have fentanyl, and these are all of the kind of like supposedly similar doses to cause this is the different amounts of drug that it takes to cause a specific effect. And as you can see, taking just a little bit of fentanyl has the same effect as taking a ton of this heroin stuff, which I didn't know it looked like that because I've never seen it before. But anywho, moving on. All right, so the rest of this article kind of just goes over like where the field is in terms of trying to work on making a vaccine. But I wanted to try to connect my little iPad so I could draw a picture of how antibodies work, because it's kind of hard for me to discuss it with just my hands. Okay. So can you guys see my fun little screen? This is my really horrible writing and my little horrible drawings. There we go. Awesome. Miss any of you? Okay, good. All right. So in this diagram, so how can we generate antibody responses to drugs like fentanyl? And in this drawing, I'm going to draw fentanyl in this green color because fentanyl is bad and bright green is a bad color. I think I just spelt it wrong too, so you can correct me later. Again, my spelling is atrocious. But as I mentioned earlier, fentanyl and other drugs like cocaine, they're small molecules, and normally when you take them, your body's immune system doesn't recognize them. So normally in your body, you have these cells called B cells. B cells make antibodies, and I'm sure you guys are all familiar with antibodies from like COVID and whatnot. And your B cells, they get born in your bone marrow. This is a very dark blue. I apologize. But inside of all of your bone, you have this center region, which we're going to call the bone marrow. And it's very protected. And inside of here, we have a whole bunch of little stem cells that we call hematopoietic stem cells. And they live in the bone marrow. They get specific nutrients and chemical signals inside of the bone marrow that allows them to grow and develop. And eventually, the bone marrow basically throughout your lifetime is creating new progenitor cells or new stem cells that will make new B and T cells and other immune cells in your body. And so when let's say this specific stem cell gets a signal that's again happening constantly, and it has done its development and become a B cell. So actually B cells, they develop inside of your bone marrow. So now let's say this is a full-fledged adult B cell, and it's now leaving your bone marrow. It's going into your bloodstream. And actually B cells, after they leave your bone marrow, they don't hang out in your blood too much. They go to this thing, they go to this thing called the spleen. And the spleen is basically this hot spot for B cells to hang out, also other T cells. And basically there's a lot of blood vessels that go throughout your body, and they drain all of the different like potential pathogens like bacteria and stuff into your spleen. And so this is also not a super anatomically correct drawing, I'm sorry. But anyways, these pathogens, they go through your blood vessels and they enter and pass through your spleen. And when they do that, there's a chance that they will come and meet a B cell. And then the B cell can potentially get activated. Now, B cells, we obviously don't want B cells to become activated super easily, because they can actually cause autoimmune diseases by making antibodies against like your own tissue, for example. And so B cells have to receive 2 signals in order to start making a lot of antibodies. Now, the interesting thing about B cells is that on the surface of a B cell, they have all of these really cool little receptors that look like Ys. And these are actually antibodies, but they're attached to the cell surface at this time, so we call them B cell receptors. And B cells are covered in these. And every B cell has its own unique receptor. So your body has millions and millions of B cells, and each of those B cells has its own like special protein sequence that makes the little receptor. So they're all unique, which means they can recognize all different types of other proteins. For example, some B cells will recognize only viruses, some B cells will recognize a specific bacteria, and some of the B cells will recognize, believe it or not, they will recognize fentanyl. They'll recognize a small like portion of the chemical structure. But, and at this point you're like, well, that should be enough. The B cell recognizes fentanyl, then it should become activated and start making lots and lots of antibodies, so that those antibodies can go bind to the fentanyl and then cause that to the fentanyl to become trapped and then eaten by your immune system. Because basically, let's say you have like, I don't know, a bacteria, and normally, let's just go over here. So you have like a bacteria floating around inside of your bloodstream, and let's say that there's a specific antibody or that recognizes it. Let's say your B cells have already become activated through the process I will describe, and they've started making these special antibodies that recognize the bacteria. What happens is the antibodies, they'll all bind to the bacteria. And this is called opsonization, which is just a really long, stupid, fancy word. And so like the special binding region of the antibody, which is like this located right at the tip of this Y, it recognizes specific proteins on the bacteria, and it'll bind to it. And then you'll have these other cells called macrophages and natural killer cells. So your macrophages, I've talked about these before. They're these big blob things, and they have these receptors that can recognize the butt of the antibody. So basically, they recognize this part of the antibody. And when the macrophage or NK cell recognizes the butt of this antibody, it'll basically tell the macrophage to phagocytose or engulf the whole like opsonized bacteria. And so the bacteria will go inside of the macrophage and get chewed up and killed. And so this could also happen. Let's say there was like a specific antibody against fentanyl or something. Maybe I should make this a different color. Let's say this is our little fentanyl antibody. Let's say that in a perfect world, our B cells have become activated to make a lot of these fentanyl antibodies. So let's say this is fentanyl. Let's say this is an NK cell. The NK cell can also recognize, and actually NK cells are not phagocytic, so this isn't a good example. So it's not going to phagocytose the fentanyl compound the same way. But let's say this is a macrophage. It'll also basically absorb and phagocytose the antibody-covered fentanyl and then chew it up and basically get rid of it, like a little paper shredder, but for proteins. All right. So I know this is a lot. Resident Evil lore again. Slashing the education budget, public education failings, ignorance, fear, gullibility. Oh, is this we're talking about 5Gs and vax conspiracies? Yes. Anywho, moving along. You almost spelled hematopoietic correctly. It's like tomato, potato. Just kidding. I don't know what I'm trying to say. Okay. Is a group of B cells called a swarm? No, a group of B cells is not called a swarm. But your B cells, basically what they do is inside of the spleen, they form this cluster. We don't call it a swarm. We call it a germinal center. So here's a close-up of the spleen. This could also be in your lymph nodes. You know, you have lymph nodes right here behind your, like, ear or whatever. When your lymph nodes get swollen, it's because all of the B and T cells inside of your lymph node are proliferating. They're literally making copies of themselves, and so they're making your lymph nodes get bigger because they're taking up more space. So inside of the spleen here, you have your little B cells, and they form like these little clusters, which we call like a germinal center. And then around the germinal center, we have your T cells. And it's very, very important that T cells and B cells are close to each other like this, because you'll find out we need the T cell to basically turn on the B cell and give it its signal too. Now let's go back to the signaling of a B cell. So let's say this B cell just bumped into and its B cell receptor bound to a fentanyl. So this is signal 1, binding to fentanyl. And I told you this is not enough to activate the B cell to make more antibodies to go attack the fentanyl. And I'm still spelling it wrong, I know. So what the B cell does when it has bound to fentanyl is it will basically internalize the B cell receptor. It'll take it inside of itself and it will chew it up. The thing is, with fentanyl, when it chews up the fentanyl, it can't present the fentanyl back out on its cell surface to communicate with the T cell that needs to give it signal too. So what will happen is the B cell will just chew it up and that's it. We have one B cell that recognized fentanyl and took it in. That's it. But there's still tons of fentanyl like molecules out there and no B cells are activated. This is a problem. So to make B cells kind of get activated, what we do is we attach a protein that can be recognized by the T cells so that the T cell can activate the B cell. So here I have drawn like this protein carrier. Basically it's a protein that's super it's very easily recognized by your immune system, and we link it to the fentanyl molecule. And this is how the fentanyl vaccine is made. Basically, they take this fentanyl analog and they link it to this specific protein, and then they inject it into the bloodstream. So this time, when fentanyl binds to the B cell receptor is internalized, and both the fentanyl and the this little protein carrier are chewed up, and they're turned into tiny little proteins. And the fentanyl is basically kind of going to go away. It's not going to get properly cleaved, but the protein will get cleaved into these little specific peptides that are going to then get loaded. So here's the peptides, the little like protein peptides from the carrier. They're going to get loaded onto this thing called MHC2. And basically MHC2 is expressed by all B cells. It's this special molecule that presents all of the peptides that have been like taken up by a cell and they present these peptides back out on their surface so that they can activate and talk with the T cells. So when a T cell, which can recognize this like special little orange peptide, protein peptide, when the T cell recognizes that, it actually gives the T cell this specific signal, which then activates the T cell. We're going to skip these other molecules because it's confusing. And then the activated T cell is going to start expressing this special receptor called CD40. And this special receptor is going to interact with another receptor on the B cell called CD40, and it's going to give the B cell a special activating signal called signal 2. And this is going to make the B cell start producing antibodies against fentanyl. And this is critical. Like without the CD4 T cell activation, without this green T cell activation, and without this special CD40L interaction here, you cannot get a B cell that will produce antibodies against anything. And so that's why it's really important to have, going back here, to have your T cells next to your B cells. Also, I just realized, I don't know if my screen has been in focus the whole time. Oops, I'm sorry. I was just looking at my iPad. Okay, going back. So anyways, the final outcome is that we get cool little fentanyl antibodies being spit out by the B cell. And ideally, these will help soak up all of the fentanyl inside someone's bloodstream and prevent them from overdosing. It'll also prevent the fentanyl from making it to the brain, where it will have its addictive effects. So people can take fentanyl at lower doses expecting to get high, but if they are also on this vaccine because they're going through rehab, they won't get the same high. It won't be as addictive, and they will hopefully be able to get off this drug. Now, I don't know what kind of withdrawal symptoms they'll have, but that's remains to be tested. And again, like this hasn't actually been put into humans yet, but the next clinical trial that's proposed for this type of fentanyl vaccine is going to come most likely from the University of Houston. There's also another research group in New York, which I think is actually where the first trial is going to take place. But yeah, if you guys are interested, I'll post some links in the chat or in Discord later. So yeah, here it is. This is a crucial interaction that happens. Anyhow, I see some questions. Spleen is a hot spot. It is a hot spot. Whenever we want to look at an immune response in the lab, what I do is if I'm like looking at an immune response in like, let's say, I'm using mice. If I want to look at how activated a specific cell is, I will take the mouse and I will take its spleen out, and then I will look at the cells in the spleen to see if they're activated or not. Actually, the spleen is full of a lot of other cells too. It's full of not just B and T cells, but it's also full of some macrophages and also some NK cells and other cells as well. It's a juicy little pouch of goodness. This sounds like it should be a Cells at Work episode. It should. I know they don't talk about this type of thing in Cells at Work because it's actually very complicated. And what I just taught you guys is something that I taught like students who were like in an immunology class. And it was a pretty advanced class. So you guys just got dished some heavy stuff, and I was a little nervous. I was like, okay, there's so much so much background I'm gonna have to give, but you know what, screw it. I'm just gonna throw you guys in the deep end and say bye. And yeah, anyways. You should write it up and make a proposal to them. Oh no, this is not my idea. This fentanyl proposal has actually been in development since 2017. And it was actually an idea that came from this lab. I'm gonna try to remember the guy's name. So there's actually two labs that I know of that are working on it. One at the University of Houston by some guy named Colin Hale. Here, I'll show you the video. And then the other guy has some really long name that I don't remember, and he's somewhere else in like New York or whatever. Let's go back to the internet. It's not going to show the internet, of course. Great. Because it hates me. Here we go. This is the little video that I found about it. So there is this paper that's actually from the second lab that is studying this. This is the Marco Pravatoni lab somewhere in the US, somewhere, I don't remember. But anyways, here they show the actual structure of the vaccine. So this is the fentanyl hapten. And the hapten is actually that little protein, that orange protein I was drawing. The hapten is located here. And then the fentanyl part is here. So as you can see, they kind of modify this part of fentanyl, which I'm going to hope and assume is the more active site that's required for binding. I'm not actually sure. I have to reread the paper and see which part of this, what this part does again. But yeah, so you can see the structures are very similar, but they've just added like this extra protein structure that gets seen by your T cells, so that the T cells can then activate your B cells to make the antibodies. Now, you can simply bypass that whole requirement for making and activating your B cells by simply just making like a synthetic or lab copy of the antibodies that recognize fentanyl. But the reason why this isn't like such a great
hello, my little coffee bean.
Jarhead,
welcome to the caffeine club.
Welcome to the stream. I hope you are well. Happy 1 year. Aw, you've been ground up 12 times. Congratulations. I hope you're well. But anyways, you can make synthetic antibodies, and in that case, you don't need to worry about activating your B cells, but it's really expensive. And so the vaccine is a much cheaper, it's the way to make it in the lab is cheaper than making the synthetic antibody. And so it would be easier to give it to people who are in like low income areas if it was cheaper. And so that's why the vaccine is more of interest than making the synthetic version. My mic is falling apart. It's like loose. Oops. Sensationalism in media? Never. No, dude, that guy was so unenthusiastic in that video. He really pissed me off. It really irritated me. I did not like it. If people use the nose spray and the vaccine at the same time, what would happen? Oh, that's a great question, Underwater. So the nose spray, I think it's Naxalone, whatever. Basically, that's let me find the actual name. I don't remember what it's called. Narcan. Yes, Narcan. I think there is another one that has like there's an X in it. That's all I know. You can use these together because the mechanism of action is different. So having Narcan and the vaccine together they won't interfere with each other, and they won't cause bad side effects. You let your asshole hang out. Wow. That's classy. Thank you for sharing. Not breathing keeps oxygen from getting to your brain. Yes, because if you don't have oxygen in your brain, then you start fainting and stuff. And also, you aren't oxygenating your blood, and so the blood going to your brain no longer has enough oxygen attached to the red blood cells, and so then your brain, the tissue and the cells are just going to get starved of oxygen and basically start shutting down. Oh, did I skip the signaling part for my students? Oh yeah, don't worry. I'm not going to make you guys learn the signaling. No, they had to know the signaling. They had to know that CD4 T cells get activated by binding through their receptor to the MHC2, and then also having this co-receptor bind to the MHC2 called CD4. That's why the T cell is called CD4 T cell, because it has a little stick that sticks out of it called CD4, and it binds to the MHC2, and that little, like that connection sends a secondary signal. well, actually, that's part of the first signal. And then there's a secondary signal that goes through another co-receptor that activates the T cell, and that ultimately, yeah. Anyways, we're not gonna get into that. It's stupid. It's not stupid. It's cool. It's not what you want to hear at 9:51 p.m. on a Thursday night. It's a quiet entrance for a change. Jarhead, you normally come in with a roar. You just kind of snuck in here. Don't insult the mic. It's trying its best. Dude, the mic is not. It's like the screw is coming out, so I have to fix it. Caff, I was being sarcastic. I seem not breathing. Okay, I understand now. Sometimes I can't detect sarcasm in chat. You guys can be sarcastic all you want, and I'm never gonna figure it out. So anyways, any other questions about, let me give you the link of this paper. This paper should also be freely available to people. It's called A Fentanyl Vaccine Alters Fentanyl Distribution and Protects Against Fentanyl-Induced Effects in Mice and Rats. So this is a mouse study. It's a really dense paper, and I don't actually expect you to read it, but I like to share my sources. Did I say that correctly? I like to share my sources with people. I cannot speak right now. Here you go. Oh wait, how did I copy and paste this? Why did I copy and paste it like that? Hang on. Why is this like this? Let me resend. What? This is a weird link. Anywho. Okay. Exit. Okay, there we go. Oh my. Jarhead. That was so loud. Okay, now you're making fun of having a screw loose. I feel called out. Okay, this whole stream is about people having their screws loosened. My screws are very loosened, okay? I'm Thank you. I appreciate it. You are not late. And actually, okay, I didn't have Milo. I was informed that it was only available in Australia. I think I can find it on Amazon though, but I didn't purchase it in advance. So instead, I made a white powder drink, and I drank that instead. I also gave you your coffee beans back, Jayski, for the redeem. So it's not like I stole them from you. So you have your beans back. Is MLA formatting an angle or a devil? What are we talking about the MLA formatting? Where's MLA formatting here? Are we talking about the citation? What are we talking about? Where's the MLA formatting? Milo is just expired Ovaltine. Oh my god. Damn it. All right. Okay, can I buy it on Amazon though? Oh my god. Okay. See, okay, by the time you guys say a joke, I've already moved on, and when I reread it, it's just awkward and confusing. Okay. So anyway, let's move on to something else. So the rest of the plan for tonight was playing some Hell Let Loose, which it's very fitting. I gave you guys some random educational topic, and now we are going to go shoot some things.